I'm Kim Mowry, Chair of the Department of Molecular Biology, Cell Biology, and Biochemistry, and on behalf of the MCB Department, I'd like to welcome you all to the Frank and Joan Rothman Commencement Forum, which was established in 1995 by President Barton Gregorian to honor Brown University Provost Frank Rothman's career and contributions to Brown University. So Frank joined the Brown University faculty in 1961, and his accomplishments were many, including establishing the graduate program in molecular biology, cell biology, and biochemistry, and earning its first training grant, which is still going strong more than 40 years later. Frank served as provost from 1990 to 1995, further cementing his legacy at Brown. Sadly, Provost Rothman passed away in 2022, but his legacy still endures, at least in part, through this commencement forum. So the commencement forum was established in 1995, as I said, by President Barton Gregorian to honor Frank's contributions to Brown and those made by his wife, Joan. Every other year, the MCB department, hosts the forum where the focus is on molecular biology, which was a great love of Frank's. Our speaker this year is Dr. Phillip Sharp. Dr. Sharp's hosts today are MCB department faculty Will Fairbrother and John Sadovy, who both trained with Dr. Sharp. We'd like to encourage the audience to hold their questions till the end of the lecture, when John Sadovy will moderate the Q&A. Now I'd like to turn the podium over there to Will Fairbrother, who will introduce Dr. Sharp.
Thank you, Kim, and thank you for coming to the Rothman lecture. I'm going to briefly introduce Phil. He was born in rural Kentucky. He grew up on a dairy farm, and he had an early interest in chemistry and math that he pursued in college, and it culminated in a PhD in chemistry at the University of Illinois. He did a postdoc in Caltech, and he was at Cold Spring Harbor before he accepted a faculty position at MIT in 1974, and he's been there ever since in many different capacities for the university. Dr. Sharp is incredibly prolific. He's published more than 500 papers during his career, and they ask very foundational questions about the nature of gene expression and the nature of genes. He's a member of many different professional societies like the National Academy, the Institute of Medicine, the American Academy of Arts and Sciences. He's won many awards and prizes such as the Gardner, the Alfred Sloan Prize for Cancer Research, the Lasker Award, and of course the Nobel Prize. And he won the Nobel Prize for the seminal work he did describing the fundamental architecture of eukaryotic genes. He's also worked extensively translating science into medicine. He's co-founder of two companies, Biogen and Amaran. And he's also very dedicated to science education, training, and mentorship. I looked on the website, and there was 123 scientists that started their graduate career or postdocs in his lab. So his lab launched many scientific careers. So without further delay, I'd like you to join me in welcoming Phil Sharp.
Thanks, Will. It's an honor to be invited to give the Rothman lecture. And if there are students in the audience who are graduating today, let me offer my congratulations. In thinking about your experience, many of you spent your first year at Brown, probably in a dorm room looking at a screen. And then the second year, you got a little bit more freedom, and you could go to some classes, but probably still looking at a screen. I'm sure in the third and fourth year, you worked very hard. I hope that's not true. I know you've earned your degree because Brown is not a place where you can not be successful. So what I'm going to do today is to talk about biological discovery as a source of innovation. And let me explain innovation. Innovation is not discovery. Innovation is not even patenting an intellectual property. Innovation is moving something from discovery through that process to the benefit of patients. And when we look at that process, we see the end outcome. We very seldom see the beginning, where the science, where the idea, where the intellectual property came from. And I will try to talk a little bit about that. Now, did I go the wrong way? Now let's try this again. Okay. I have some conflicts of interest. Biogen, as was mentioned, I co-founded. I was on the board for 29 years. I am no longer associated. Alnylam, I am still on the board of, and DuPont is a very new venture which I advise. So let's talk about molecular biology. Molecular biology really had its origins in the work of Francis Crick and Jim Watson when they proposed the structure of DNA with data from Rosalind Franklin. And this was the first time that man knew the chemical nature of inheritance of information between generations. It was a signal event in biological science, and it was a signature event in launching of molecular biology. I know both of these individuals, have known them. Crick was an analytical scientist, very, very critical. Watson is an intuitive scientist, very intuitive about what's important. And the combination of the two were able to give us a structure of a gene. And that was really the launch of molecular biology. That happened when I was in Kentucky, here as a 9-10-year-old person on a farm. Nearest house is seen behind me. And I grew up there, but I can tell you that I didn't hear about DNA until I was a senior in college. So for about 20 years, it was a very small community. Molecular biology was of great interest to that community, but there was really not a lot you could do with molecular biology as a discipline that can benefit people and be innovative with. However, I'm doing something wrong. However, there were great advances. About 10 years post the discovery of the structure of DNA, this diagram was published by Jacob and Monod. And what is shown in this diagram is that helix at the top, which is DNA, a number of genes, Z, Y, and A, are designated there as segments of that DNA that codes for those specific genes. And you see below the DNA, there's the message RNA that is copied from the DNA in the sequence of the DNA. And that message is decoded into making specific proteins, in this case, 3 proteins that allow the metabolism of a sugar. And therefore, you see the information flowing through nucleic acid from DNA to RNA to protein. And you see to the left another gene, I, that encodes another protein, a repressor. And that repressor turns this copying of the RNA off if the sugar is not present. But when the sugar is present, it then is inactivated, you express these genes, and you get the utilization of the sugar. So this is regulation, this is messenger RNA, and this is protein being encoded by messenger RNA. And you see this very nice set of sequences that code for a given protein all being continuous. Well, it took us 15 more years before we understood how your genes were structured. The structure of your genes look like this. This is a strand of DNA, as shown here. You see those black boxes that are called exons? In those exons is a sequence that makes a protein. And what happens in your cells is that these sequences are spliced together, and these sequences are called introns, are excised and degraded. So that's your genome. It's that long. It's about 3 billion base pairs. 2% codes for protein. That's your functions. The 98% is in introns and between genes. And most of that is still dark matter in terms of genetic material. Now, you have 23,000 genes, all of you, 23,000 genes. Worms have more genes than you do. But you have something called alternative splicing, where from one gene you can make a variety of different proteins. So for example, here's an exon spliced to this sequence, and that makes a protein that has all three of these blocks of sequences. And this one splices alternatively, leaves this out, and makes a different protein. They're related in function typically, but they're different. So out of those 23,000 genes, there's a large variety of information used to make different functions. And it's particularly prominent in your brain, where this type of splicing can be much more elaborate than just the one example. Now, something else happened in the mid-70s. That discovery was in '77. And that was a series of technologies that came from academic scientists wanting to understand the function of a gene, the structure of a gene, and how a gene worked. And in 1972, we learned how to take pieces of DNA and join them together. That was called recombinant DNA. In '77, before it was published in '77, we knew how to do this. Before that, we could sequence DNA. Very labor intensive, but we could sequence it. And in the '60s through the '70s, we learned how to chemically synthesize it. So here came together in the mid-'70s the striking ability to do synthetic biology. We could make new genes. We could move genes between organisms. We could make new organisms. Synthetic organic chemistry was 100 years old. This is the first time we could do synthetic biology. And that led to a lot of interest in how to take this technology and actually make something that is useful. And I was involved, as I said at the beginning, in starting Biogen with a number of other scientists in 1978. But the first biotech company that came out of this recombinant DNA was started in 1976 on the West Coast with a company called Genentech. And Genentech took this technology and made the insulin A and B chain. It made other genes that are used for therapeutic activity. Biogen started in 1978. Interactions between venture capitalists and myself and Wally, and then a number of other European scientists, led us to form a scientific advisory board, develop funding from venture capital, and ultimately from others. And that led to the founding of Biogen, originally in Geneva, Switzerland, but then subsequently in Cambridge, which I'll show you in a moment. What was our motivation? Our motivation at that time is molecular biology had been a discipline that developed from '53 to '77. There was no private sector activity in molecular biology. And the idea was this science is going to make a number of very useful innovations We needed to develop a private sector to fund this, and that started the company. So Biogen now is a very large company. It's one of the oldest, it is the oldest freestanding company. The interest initially was in medicine and energy and in chemicals, all of which ultimately, much of which will be made by biological systems ultimately when we run out of hydrocarbon, which we're working very hard to do. So in 1977, this is MIT here. This is Main Street in Cambridge. MIT is to the left. This is the Kendall Square area, and you see a parking lot. Biogen's first lab was over there on the other side of the parking lot. And now, this is 2012. MIT is here. This is MIT to the left. This is the same region you just saw. There's Draper Labs right there. And it's now, in 2012, developed space, jobs, scientists, taxes, income. So it's developed enormously, and it's become the most dense biomedical research community in the world. So this is a diagram of the environment of MIT. MIT is these red buildings here on the Charles River. And what you see here in these greenish boxes are significant size biotech companies located in Kendall Square around MIT. There is about 100 of them, but some of them are very small. They can be 10 people, and some of them can be hundreds. And then you see some large pharmaceutical companies who have moved their research labs into this space. Novartis, Sanofi, who bought Genzyme, Pfizer is over here. Biogen is there, and the company I'll talk about in a moment, Alnylam, is here and here now. And now let's talk about the Broad Institute, which is associated with MIT and Harvard. And it was started by Eric Lander in 2023. So the Broad Institute was the signature event in the Cambridge area in bringing genomics to molecular biology. Now, I told you in the 1970s we could sequence DNA, but could we sequence 3 billion units of DNA all in a line and then understand what it meant? And Eric Lander and others launched the Human Genome Initiative in 2019. The first sequence of the human genome was reported in 2003. And that's a sequence of a large number of people that gave a consensus structure in the number of genes and other activities. But it has been the signature underwriting event of most of what molecular biology and cell biology has been involved with since. So it took about 50 years to go from the discovery of the structure of DNA to being able to sequence genomes. And we've sequenced genomes across many different organisms. So here's the striking advances in technology over that time period. In 2001, it cost you about $100 million to sequence one genome, one person's genome. And this is the drop in price at $215. Right now, you can sequence a human genome at a level of detail that can be used to diagnose genetic diseases, identify single variants in the genome at about $100. So many people have had their genome sequenced. I won't ask, but many have for medical reasons, and there are many reasons for that. But if I take genomes from you and sequence them, I will typically find variation between you at 1 in 1,000 positions on the sequence. So that says that you vary between each other. Most of this variation is totally neutral. It has no effect on anything. But it is a variation in the population because DNA changes over time. And you can use that to make lineages and direct identify DNA to certain pieces, certain people. So $100 genome means a lot more people are going to be sequenced. So what do we use it for? Well, one thing particularly, we use it for cancer because most cancers are caused or have at their core a mutation in genes that are called oncogenes that make the cell proliferate and change, or lost breaks to that process called suppressors that inhibit those changes. And you treat patients dependent on those sequences in part. But you also, and what this shows you, is the number of mutations in the cancer cell that differ from the variation in the genome. And if you have cancers that have experienced a great deal of mutagenesis, such as having a defect in repair, smoking, sunlight, and melanoma, they have high mutation variation. And that means that you can frequently, about 50%, treat that patient with immunotherapy and get very long-term survival. So these tumors that have lower levels of variation only infrequently respond to immunotherapy. And these are pediatric cancers that have almost no variation except in those genes that are driving the cancer. But there's another use of this sequence that I find more interesting, and that is the genetic basis of disease. This analysis or summary was taken from 2015. At that time, this is from a group of human geneticists. We knew 3,000 different genes that had mutations in them that we know cause variation in the adult that segregate by Mendelian genetics from parents to daughters and sons. 8% of all live births have a genetic disorder that are identifiable by changes in phenotype when they're young adults. If you have a child with a genetic disorder, severe one, over the lifetime of care, that's about $5 million worth of care and loss of contribution. And the striking thing is the diagnostic rate of these genetic disorders. Children, it's about 1 in 10%, and in adults, it's about 1 in 3. So there's a large fraction of genetic variation that we know causes phenotypic, in many cases, diseases that are not diagnosed until the patient has progressed to a point of losing function. Cells have died, and once cells have died, it's very hard to reverse that. So intervening early is a major step forward in treatment of any disease. So let's talk about some treatments of diseases with RNA and DNA as the basis of those. And I love this picture. This is Emma, and Emma can walk. Emma, probably if she'd been born 5 years earlier, wouldn't walk. She might not be able to survive over a year, maybe 2, because she has spinal muscular atrophy. But because she could get a treatment, she can now walk, and she looks so happy. I just love that smirk on her face. So what is spinal muscular atrophy? Well, it's a genetic disease. There's a cluster of genes. You see SMN1, spinal motor neuron 1. Lose that gene, you lose the function of spinal muscular neurons, meaning neurons that control your muscle, and that is the basis of that SMA disease process. You see this gene is only diagrammed here with one exon, but that's a gene at the top. And patients who have this disease lose this gene. This gene is spliced 6, 7, and 8. These exons are spliced together to make the functional protein. Now, you should know exons now. I told you about exons. I told you about messenger RNA. And it makes a full-length protein that's totally functional. If you have this gene and no splicing, you do not have SMA. But if you lose this gene, there's a related gene next to it called spinal muscular neuron 2. It is not spliced 6, 7, and 8. It splices 6 to 8, and that produces an unstable protein and doesn't do you any good. The protein's degraded. So if you could turn this gene onto making that splicing pattern, you wouldn't have the symptoms of SMA. And the way that was done by a collaboration, which I'll explain in the next few moments, is Adrian Craner at Cold Spring Harbor identified sequences in this intron that are inhibiting the use of that exon and making 6 jump over to 8. Developed an ASO with Frank Bennett at ISIS that, when injected into the spinal column, would enter neurons, block this protein, and shift the splicing to 6, 7, 8. First treatment of splicing. Splicing was discovered in '77, and it then took about 30 to 40 years to get a method of modulating it. We now know some small molecules that modulate it. We now are using gene therapy to modulate it. This was the first modulation. And it was the product of a lot of very fundamental science, who I should acknowledge. Adrian Craner at Cold Spring Harbor developed the science on SMA. And in 2008, he started collaborating with a scientist at Ionis. And that company was started in 1998. And this was its first major important advance. And Biogen licensed it in 2012, and it was approved 4 years later. So from 2008, when the science was done, it still took 8 years to go through development and get the product. And that's biotech. So that was fundamental science leading to therapeutics, innovation. About the same time as this was going on, there was a major discovery that no one anticipated in 1998. And that discovery was done with a simple worm that we grow in the lab, and it reproduces very rapidly, and we can genetically manipulate it, and it's transparent so we can see each cell in it. It's a wonderful experimental organism. It's called C. elegans. And Andy Fire, there, at the far right, and Craig Mello were interested in activating a gene in this organism. And I won't go through the experiments that turned into a puzzle to them, but they ultimately discovered that if you take the sequence from a gene in the form of double-strand RNA, Now that messenger RNA is a single strand. In this case, the RNA is duplex, it's double strand. You inject it in the worm and it'll inactivate the gene. And that was RNA interference in 1998. And in 2006, Farr and Mello got the Nobel Prize. Now, I have a particular relationship with Farr. He was a grad student in my lab. He showed up with a hairdo like this. He was 19 years old. He'd already graduated from Berkeley in mathematics. I think he had one course in biology. And he showed up at MIT in the biology department, and he came to talk to me. And this man was interesting. He was very quick. So we took him in the lab. He did biochemistry, pretty boring, straightforward stuff. But then he went off and made this discovery at the Carnegie Institute, and magnificent scientist. He has another discovery, won't tell you, it's not out. But in '98, I learned this from Andy Farr in a publication, talking to him. At MIT, myself, David Bartel, Tom Tushel, Phil Zamar, and ultimately Schimble was later, we worked out the biochemistry of that process and discovered small RNAs, which I will show you about in the next slide. And we established in 2002 a nylum, which on that screen when it was announced or described by Will, it said unknown. But it is the brightest star in Orion's belt. That's a nylum. And it's a string of stars, and the brightest is a nylum, and that's where the name came from. It came from Paul Schimble. The company was established 4 years after the discovery, just as the biochemistry was done. And it went public in 2004, and its first product was approved in 2018. 16 years after the founding of the company. But it was a whole new technology. It's a new way of potentially treating disease. And once you discover it, it'll never disappear, as long as man can read. So what is this technology? Well, it's taking small double-strand RNAs, which is called short or small interfering RNA. These are RNAs with 2 strands, and introducing them into a cell, and it'll result in cleavage of the message. So we know DNA makes messenger RNA, which is then used to make protein, and this RNA, when introduced into the cell, will cleave this message and silence that gene. Think about that. I have the sequence of the human genome. If I can make a synthetic RNA 20 long that can be introduced into cells, I can literally silence any gene, any place, if I can deliver it. That's the issue. But it was very interesting, and well, could it be useful? So we now know at the same time in around 2000, and I won't take you through this pathway, but you have 500 genes we didn't know about. We couldn't identify them. Those are genes that make these small RNAs in your cells from your genome. And they are siRNA-like, and they go into a professional complex of proteins that pair them with messenger RNA and will lead to degradation if that pairing is exact. So we had this whole pathway. We didn't know about it until 2000. So what was the prospect? Can we take this discovery and make innovation out of it and harness a natural pathway? Well, the big issue, and we'll talk about more of that in a moment, is how to introduce these small RNAs into cells because RNA is very water-like. And the surface of this cell, your cells, is an oil. And water and oil don't mix, and RNA does not pass through that membrane frequently. So it's a catalytic process. Once it's in the cell, we know how it gets loaded into a complex, how one strand is taken, how it's paired to the RNA and cleaved and recycled. We know all that. We know the sequence. We know the sequence of the gene. But can we get this RNA into cells? And that was a major challenge of the company. So how did we devise first to get it into cells? We created a virus particle. It's called a lipid nanoparticle. It's shown here. It's got a membrane. The RNA is packaged internally. It's got glycopeg on its surface to keep it in liquid. It binds to a protein circulating in the blood, and that targets it to receptors on the liver. It's internalized through those receptor interactions. The pH turns from 7 to 5. The lipid changes structure and releases the RNA into the cells. Had to improve the efficiency of this by 1,000-fold, and Peter Tullis was the first to recognize that these cationic lipids were important for delivery of RNAs to cells. All of you have been treated with this, essentially. If you got vaccinated for COVID with mRNA, this is the lipid nanoparticle you got vaccinated with, or a similar chemical structure. So this first treatment with siRNA was for a genetic disease called transthyretin-mediated amylosis. And what that means, if you have a mutation in this TTR gene in your liver, you will make a protein that misfolds, accumulates in your nerves and your muscles, as shown here by the green in a pathological slide. And it typically onsets at about 30 to 40 years of age, and it typically takes 5 to 10 years before you die. What was shown is that if you treat with those lipid nanoparticles, you can see silencing in the liver. This is patient bloodstream TTR levels. This is placebo, a non-relevant RNA, and you see silencing for about 2 years or 3 years. And patients have been treated with this for now almost a decade, the early patients. Now, that's an example in which you take fundamental science discovered around turn of the century, turn it into a product in 2018. Now there's 5 approved products. The Onpattro is the one I described. Mutra is for the same TTR but wild type. This is a porphyria, heme porphyria that causes extraordinary pain in women if they have a genetic disease and it accumulates. This is oxaluma. This treats patients who have high circulating oxalate, which makes kidney stones. Okay, and in children, if they have that condition, they can lose their kidneys within the first year of life, as I will tell you. And this treats cholesterol, PCSK9. So it took us 10 years, 16 years to get to this stage. Let me talk about one example. This comes from Alnylam just was, and it's more busy than it should be, but I didn't get a chance to simplify it. Here's a 30-year-old woman in southwestern US who lost her first child to hyperoxalemia primary, and that is this oxalate condition, 11 years ago, and has since become knowledgeable about the disease in the PH1 community. In 2023, she became pregnant with her second child, was diagnosed with PGH1, the child was, in utero. Her physician reached out to Alnylam and made the arrangements to have the child treated. The baby was born in early 24 and started treatment within an hour. They arranged for the treatment. The Alnylam field representative helped. The baby is doing great and is scheduled for his third dose in early May. So there's a number of patients being treated by these diseases. They have no other hope at this moment. There will be other treatments, and they'll be more convenient, maybe more effective, but this is an important treatment. So, where's this going? I don't really know. I suspect it'll be a very useful treatment because in one version of this process, you can get treated with RNA for one time every 6 months and have sustained silencing. Some cases it might be every year. So if we just have to wait and see how patients respond in diseases. But the most exciting part for me is, can we do this in the central nervous system? Because we know there's Alzheimer's, and we have our first treatment for Alzheimer's that slows the progression of disease. That was by Biogen, and it was a couple of years ago, and it's now approved. But it's certainly not the ultimate treatment you would like, because only 30% of the patients benefit from it. Parkinson's, we have essentially no treatment other than for symptomology. The disease progresses. Frontal temporal dementia, Huntington's disease, ALS, cerebral ataxia, many others. The brain is a very difficult organ to treat disease because A, we don't know a lot how it really works because it's a very complicated organ, and B, we have to get drugs across the blood-brain barrier to get them into the system to actually have an effect on the disease. And these are injected in the cervical spine and spread throughout the CNS. And this is an experiment where an RNA to SOD1, which is involved in ALS, was injected in this lumbaric region in rats. And then over time, given spread up through the system, the experiment was done as well. How far can you see genes silenced in this system? So you go to the cervical region, you get 90% silencing, thoracic, lumbar, cerebellum, up here. Prefrontal cortex, not as much, and the remaining brain, about 50%. So it would be wonderful to be able to impact on those CNS diseases. Now, I'm not going to take much more of your time. But has discovery stopped? Are there frontiers? Do we know everything? No. Surprise. We've looked at cells for the last 150 years, and we've been able to see in the midst of cells membraneless bodies that are condensed with proteins and RNA and DNA, and they're all concentrated there. They don't have a membrane around them. They're concentrated by another type of interaction. And what was surprising, it was discovered that not only are the proteins and DNA concentrated, but they have liquid-like properties. What does that mean? That means that if you press a shear on it, they distort like a liquid. If you measure the rate at which they're exchanging proteins with the surrounding solute in the nucleus or cytoplasm, they exchange proteins very quickly. They're very dynamic. And it's that dynamic property that makes them quite interesting and requires a certain type of interaction, high valency, high multiple interactions, but fairly weak interactions. And that concentrates these proteins in different structures. And when we started looking for them, we realized that they were involved in the transcription, that's making RNA from DNA, the splicing, that's in the speckled regions. The DNA repair uses these, makes transient condensates. They're involved in cell division, and they're involved in synaptic activities and how our brain functions. So this was basically, from 2017 was this. It was a little earlier when the first paper was published. But it's a whole new way of looking at most cellular processes. A type of interaction we had not appreciated how fundamentally broad and important it was. So with these insights, can we turn this into understanding diseases better and controlling them and giving people a longer and healthier life? I don't know. It'll take years. It'll take investment. It'll take people working very hard and honestly and publishing their results and promoting the field. But it's an adventure. I'm associated with DuPont, as I pointed out, that's doing this, but I'm not a founder of it. So it's been, for the students in this audience, this has been a totally fascinating life. It starts with an education. It starts with working with very bright people doing really interesting things at a fundamental level. And then taking the time out of that life when the opportunity comes to try to help take that technology and make it useful for people. And that conjunction, I've been a scientist at MIT for 50 years. I've run the Department of Biology. I've run the Cancer Center. I started in neuroscience. I teach every year for 50 years. I love it. That's why I stay there. I love it. But I do think it's very important to have the opportunity and reward people and celebrate people who take and work very hard at taking these discoveries and working through everything that has to be done and testing it in patients to show that it benefits people. And that's what it's been about. These are people who've worked in my lab over those years. This was my reunion in 1994, and in 2004, these are people who are postdocs and graduate students and undergrads who worked in my lab, and I've learned as much from them as they've learned from me, and I've stayed in touch with most of them, and it's really one of the joys of my professional life. So thank you for giving me the opportunity to give this talk, and I would be pleased if we have time to take questions.
I'm John Sedovey. I'm going to moderate the question period. I'd like to mention at the outset that we're going to have a reception immediately afterwards. And all of you that were at the Horace Mann lecture earlier today, this reception is for both of those lectures, so please stick around. I was going to say something about your lab, Phil. When I started in 1984, I was given the furthest bench in the lab. And it was a horrible mess. It was Andy Fire's bench. It took me half a day to clean it up. Please, sir, you had a question.
Do you have any views on applying this to long COVID?
I do, and I don't. That's what it said. I do and I don't. The major issue of taking this to long COVID is we don't have a biomarker. We don't have markers of that disease that tells us really whether it's an immune disease or problem, or whether it's a amyloid problem or other problems. And without such a biomarker, it's very hard to know how to begin to think it. You got to be able to track the progression or the degression of a disease, and that's where I'm at. I am an advisor. I just stepped down from it to a company called Vir that developed COVID antibodies and small RNAs to hepatitis B. So we looked at that issue and just didn't feel that we had the basis of science to be able to do it. So we need science. We need a better understanding of the physiology of those conditions.
Question here, Phil?
Yes.
Hi, so we've evolved over millions of years to fight off exogenous nucleic acids in the form of viruses and other pathogens. Have you been surprised at how easy it's been to get these siRNAs and other nucleic acids into cells and do their thing?
We've known for a long, long time that you could get DNA into cells by putting it in very particulate matter, and very inefficiently it's taken up in the cell. And as I pointed out, we've designed these lipids to be able to fuse the membrane and go into cells, or use a targeted receptor on the surface and very inefficiently get into cells. I wouldn't say how easy. That cost over $1 billion. Okay? And a lot of really people hard work at it. Now, I have an ex-undergrad who worked in my lab at MIT, went to Stanford, went to Berkeley, has come back at Harvard, and he's found RNA on the surface of cells. And it's glyco-RNA. It's modified by glycolysis. For the rest of you in the audience, you won't know that, but what that means is a sugar on it, and the sugar means it has a pathway out, and it may even have a pathway in. And so I'm very optimistic that we may find a source of communication in biological systems, in our systems, that is RNA moving. I wouldn't be surprised.
Phil, there's a question back here.
There's one over here. And then we'll come over there. Yes, please. There are two mics, John. Yes.
We found that
Hold your mic up, please. Oh, I'm sorry. I didn't know he didn't have a mic. Let's go over here and then we'll do it. Go ahead.
You
Yes.
spoke about mechanisms once the reagent is inside the cell, and you mentioned how you can get that inside the cell. Could you speak to how you can target cells specifically and how people might develop that technology?
Well, that's where the field is at. These lipid nanoparticles, they're a certain size and they have a lot of surface. And I think it's going to be a challenge to be able to target them other than deliver them locally and get high concentrations, which is what the vaccine does. But the other techniques, that is tagging the RNA with a chemical that has a receptor on the surface of the cell that gets internalized. That is the way the second process of delivery that Analem's developed, and it's developed for the kidney right now. But they also have a process for the brain now, and they're broadening their research here. I think that technology will be more targetable, more convenient, and is something that a lot of other companies as well as Alnylam is investing in. It would be good to be able to get it a little more efficient. But targeting specific cells can be used in many therapeutic treatments. So as that problem is solved, I think it'll also impact on how to get specific tumor cells targeted in other cells. Please.
Yes, it seems that the COVID virus keeps mutating, and it's hard for even the mRNA vaccines to keep up. Do you have any thoughts about having a vaccine that would, would deal with the eventual variations of the virus, or is that kind of a hopeless possibility?
The issue, okay, if you can take a variant and sequence it, and that can be done in a day, and then you could make RNA that would contain those sequences as part of a vaccine, and that could be done in a month or two. And then the question is, does it work and is it safe? That takes time. Right, because you have to find a population that's at risk and then vaccinate them. But before you do that, you have to show that the RNA doesn't stimulate autoimmunity, various other issues. And then once you've done all that and you've shown it's effective, then you can get FDA release, 6 months. The variant has disappeared in many cases before that happens, right? But the good news is that if you're vaccinated, and I've been vaccinated 3 times, and then if you get infected, and I've been infected 2 times, you really develop a good immunity. And I don't worry about COVID anymore, even though I'm quite senior. But it is true that as this virus has moved in this type, even though the surface is, the neutralizing antigen varies, if you've been experienced with vaccination and possibly infection, you'll get more resistant to it.
Question here.
Hi.
Yeah,
Hi.
Hi.
When you think about the pace of development and breakthroughs that have happened over the past few decades, you started out in the 60s and the 70s and where we are today, and you apply that to the future, where do you land when it comes to those neurodegenerative diseases that you listed for which there still remains to be dark matter?
10 years from now, we'll have a lot of control.
Thank you.
But in a broader sense, I didn't have on that list schizophrenia and bipolar and manic depression and other things that are emotional or psychotic states. Those are prisons. If you become schizophrenic, you have a lifelong limitation as to what you can do. So that and the issue of the brain is a frontier. We're beginning to make progress in understanding it in the sense that you understand it with chemical specificity in different cells. We're just at the stage of being able to pluck cells, one cell, out of the brain and ask what's going on in that cell. All right? So I think we'll have more impact over the next decades. I put that out at 2 decades, not 1 decade, in that, being able to impact on those issues as well. So not with this technology. I'm not optimistic. I don't know. I have no idea how it'll all be. The science is just beginning to start. So it's a frontier. It's a very important frontier. It's how we learn. It's how we think. What is that going to mean for us? Are we going to exceed AI, or AI is going to exceed us? Don't know, but it's going to be interesting. Yes,
Phil, you charted marvelously, the path from your discovery of splicing to then we have Spinraza that cures SMA. Yeah. And then you are at the origins of RNAi, and now we have the RNAi treatment for TTR. Are you surprised by how well it went? Like when you were looking at RNA dynamics in the 70s and 80s, and here we have all these medicines, are you surprised? Did you know this was happening?
I can't say I knew it. But I was fascinated by RNA, right? Once I got thinking about what a gene in our cells was like, and then there was hints that there was something strange going on, very long RNA in the nucleus and short in the cytoplasm. And that led me to do the work that led to the splicing discovery. And then as, my colleague, not my colleague, he's a colleague in the field, not he trained with me, Tom Check, discovered catalytic RNA, and that RNA could actually carry out enzymatic reactions. It was Sid Altman. It's empowering, but Andy Farr, when I read that paper, I almost fell off my seat. Here was something I understood, that's small RNAs, and it could silence a gene. And and combined, as I said, with the known sequence of genes, it offered a normal pathway in manipulating those genes. I've been looking for that for 30 years in mammalian cells. And so I would have bet anything, and it did. I mean, we just started the company. We didn't know what we were doing, but we got some RNA chemists, and we got some physiologists and medical docs, and just started working on it. Last question. Oh, we'll take two. You got your arm up. We'll take two. Two questions, I'll let you go.
The therapy you talked about turned off genes or parts of genes that were making bad stuff.
Yeah.
Can you say something about the genetic disorders where the gene is not doing what it should be doing at all, sort of the opposite disease, a gene function that's missing as opposed to a bad gene making stuff that caused trouble?
There is work going on, not necessarily with Alnylam, that is looking at ways of taking this technology and doing what you're saying, augmenting the level of a protein being made from a given messenger RNA. Whether that'll pan out. In most biological systems, everything is coupled to a feedback. If you got something going here, you got something inhibiting it, so it doesn't go too high and cause damage. And frequently, you can actually inhibit the inhibitor and get more. And that you can do. But if the gene is missing, then you're going to do gene therapy. And gene therapy is changing rapidly, not because the technology's changed a lot. What's happened is people are accepting it. And that then is developing a whole new approach. Spinal muscular atrophy, I was told, is frequently treated now by gene therapy in the CNS. So there's ways of turning on. Messenger RNA turns on. It's a positive mode, not negative mode.
Hi. You've spoken about your students, and we see many of them here, and postdocs and whatever. So, and over the course of your long career, do you have any observations about what makes a scientist? What are the qualities that result in someone
I've
being
had
productive?
scientists who are absolutely brilliant. I've had scientists I didn't think was so brilliant. I've had scientists who work 12 hours a day, and I have scientists who work 2 hours a day. Some are broader, some are narrow. You know, it's finding that magic that just makes things work, that you know, is the secret of success. I will say this. When people have a broad interest and curiosity about biology and then biochemical systems and molecular biology, that breadth with, you know, training and commitment is really an important attribute. And the other is ability to work with people. Ability to work with people, because you can't do things by yourself. Not only you can't even think of things by yourself. You need people to give ideas to you. You need to be in an environment in which you have open discussions where you're criticized and they're criticized and everybody knows it's goodwill. It's trying to get something done, and you know, and giving them their space to make their mistakes and space to make your mistakes, because it's out of those mistakes that you usually learn. I want to thank all of you for your attention. Thank you very much.